Well, welcome everyone to our session today on maximizing efficiency in your lab. Our goal for the session is to go over tips that will help you stay organized and effectively share and collaborate with all of your teams. And just a couple housekeeping items before we jump into things here. First, if you have any questions, feel free to throw them into either the chat or the Q&A. And when you do, remember to address the question to both panelists and attendees so that everyone can see what you're asking. Likely, if you have a question on your mind, there's someone else that's thinking of the same thing as well. So feel free to ask whatever pops up. And then you can use the chat to say hi or chat with others in the audience if you'd like as well. I'm actually joined by my colleagues, Kat and Mayette, who will be keeping an eye on the chat to make sure that any questions that come in will get answered. And we don't have a dedicated Q&A, so as questions come into your head today, feel free to just ask whenever. We want to make sure that you're getting the most from this session today. So if there's anything that's unclear, anything that you want to dive deeper into, feel free to let me know, and then we can walk through it together. And we'll be together over the next 45 minutes or so, and I thought it'd be nice to introduce myself and not just be a floating voice here. My name's Tim, and I'm a science communication and customer success manager here at BioRender. I'm a scientist by training. I did my undergrad in biochemistry before getting a PhD in pharmacology and therapeutics. And it was really during my time in research that I experienced many of the challenges that all of you likely face on a day-to-day basis. Things like it being difficult to communicate complex ideas and workflows without a common visual language. And you probably all experience this as well, but you know it can be difficult to communicate results to people outside of the folks that are immediately working on a project, whether it's across the teams and departments, upwards towards scientific leadership so that they can clearly understand what is being done in the project, or even for new folks that are joining the lab, it can be difficult to onboard them to like the processes, the concepts. So there's a lot of it's not always the smoothest route of communication. And speaking for myself, I also found that I often got very disorganized. Some of you might be better than me at this, but what I found was that a lot of my files just ended up being in one big folder with everything in it. And then I would spend a lot of time trying to find the right file to use at the time. And lastly, what I noticed was that it was inefficient trying to collaborate and share feedback because it felt like things were just all over the place. You know, comments were in one place, the actual work was in another, and it became difficult to try and bring all of that together. And that's why I joined BioRender, because I wanted to help scientists avoid the same problems and struggles that I went through and show people that there's an easier way to do things. And what I wanted to do was help folks, get next steps approved faster, help you surface ideas to different teams a lot easier to really foster that sense of collaboration, and also help you get easier project approvals because then the people that you're communicating to clearly understand what is being done in the research. And we're able to do this because BioRender helps us standardize these images, the images that we create to represent our research, and it allows us to clearly convey our message visually. And so here on the slide, we have some examples of illustrations that were created with PowerPoint, with Illustrator, and this is how I think many of us are accustomed to creating our figures. But the challenge here is that you can see the diversity in the images that are created. However, they are all communicating the same concept, and that's that they are antigen presenting cells interacting with T cells. And when you have such a large discrepancy, that was really that spark that drove the founding of BioRender, because we wanted to standardize these and start to level out that playing field and make it so everyone can clearly convey their message visually. And really, our goal is to accelerate the world's ability to learn, discover, and communicate science. And for today, what we're going to do is actually look at 3 tips that can help maximize efficiency in your lab. And these are team organization and file management, how you can create visuals to communicate your research, and then also looking at team collaboration and feedback as well. And again, we don't have a dedicated Q&A today, so whenever you have a question that comes up, feel free to throw it into the chat and Q&A. Again, I am joined by my colleagues who will be keeping an eye out on both of those channels to make sure that we are able to get to your question. All right, let's get started here with our first tip, which is how we can optimize team organization and file management, particularly when it comes to your team's illustrations. And I think often we spend a lot of time looking for specific files or looking for the right file to share with our team. And I think all of us have probably been here or at least approached this. Um, but I think all of our desktops have looked like this at some point or another. Maybe not as bad as this, but you get the gist. And I'm sure all of you remember that when it got to this point, trying to work on a file or trying to find something to share with someone or make changes, make a copy, send it back, all those little things took a very long time. And that's because it felt like the file that we were looking for was never in the last place that we left it. And this can be very inefficient. And it's the same with our BioRender gallery. And as you use BioRender more, naturally, you're going to create more files and things might start to get a little bit disorganized. This is actually a screenshot of my gallery. Yours probably looks better than this, but once it starts getting to this point, you notice that a lot of your files start getting lumped together. You have things that don't have the most descriptive file names. You can see that a lot of my files are simply called untitled. And that's not the most helpful for trying to find an illustration, especially if you have more. Or on the flip side, you have files that just read as copy of copy of copy of untitled. And again, that's difficult to parse through when you have a lot of different illustrations that are floating about. And just like with our desktops, when your gallery gets to this point, things become inefficient. It's harder to find your illustrations, harder to share your illustrations, and things just kind of slow down. And instead, what we can do, and let me hop back out into my gallery here, is we can develop folders and we can use different features to help us stay more organized. So right here, right now, you can see that I have my unsorted gallery here, a lot of different illustrations. And let's say that I am working on a project and I want to house my illustrations within it, all like the relevant illustrations in it. What we can do is actually create a folder for ourselves. And you'll see that I've created some folders already to help stay a little bit more organized. But what we can do is create a new folder, and let's just call this one, project progress meeting. And then we can create that. And you'll see the folder pop up here. And then to just move our files, we can just drag the ones that we need to move and then just pop them into that folder. And we're going to drag this one as well. And when we go into the folder, all of our illustrations are here. But if you make a mistake, you put something in there that shouldn't be in there, it's as easy as just grabbing that file again and then putting it back into the right place. And so for this one, I'm just going to throw it back into my files just to show that it's really easy for us to shuffle around the illustrations to make sure that we have the right files in the right folders. And as a heads up, everything that you create is going to be private by default. And so any files that you create, or any illustrations, any folders, those will be only accessible to you to start. But oftentimes I think your colleagues will want to access your illustrations. Maybe you've created something that they'll find helpful, or maybe that they've created something that you'll find helpful. And a story that was shared to us by a scientist that works at a large pharma that we partner with, he shared the story where when someone goes to create a PowerPoint presentation, they remember seeing that, oh, so-and-so had a figure that would be really useful in my deck as well. So what they do is actually go and find that deck, take a screenshot of that illustration, and then bring it into BioRender to try and make those changes, even if that figure was originally created in BioRender as well. And I'm sure I'm preaching to the choir here, but you all know that trying to edit and modify a static image is very tedious. It becomes very patchwork at the end, trying to, censor parts of the illustration that we don't want to see, or trying to make some creative edits to it. It's very hard on an illustration that doesn't move. So instead, what we can do is actually share illustrations within BioRender so people are able to easily build off of it as a template. And I'm going to show you how we can do that. And so we can either choose to share individual files, but more often than not, there's going to be multiple figures that pertain to a certain project. So we can also share folders as well. The process is going to be the same. I'm just going to use folders as my example. But what we can do is just highlight the folder that we want to share, click on these three dots, go to share. And from here, I can start to enter in the names of the people that I want to have access. So let's say that I want my colleagues that are here today to have access to this as well. So I'm going to invite Cindy. I'm going to invite Mayette. And I'm going to invite Kat. And now everyone that I've sent this to can now access this folder, access the illustrations within it, and be able to make edits to it. But this is just with 3 people. And you can see that as we have more and more people within our lab or within our team, having to type out the names of every single person can get kind of tedious, especially if you need to share multiple folders with people. This can start to add up. So what I'd recommend doing if you have a lab that you normally work with, like with some core members, what we can do is go up to our profile on the top right here, and then go to groups. And here in groups, we can actually create different groups that include all of our lab mates. So I can repeat the same thing. I'm just going to call this my lab, and then create that. And just as a heads up as well, as you're adding members, you'll need to add yourself in as well. So I'm just going to add this here, and then we can add in more people to it as well. So I'm going to add the same folks. And this way we can start to build out our lab, and it makes it really easy for us to start working on illustrations together. And if needed, you can also add in a custom message to send along with your invitation. And then what we can do is add in all of our members. And you'll see here, there are 4 people in our group now. And if I go back out to my gallery, if I need to share more, if I need to share another folder, I can just click on those 3 dots, go to share. And then when I click on the text bar here, I can actually just select my lab and then send all 4 people an invite, and now they all have access to my to this folder as well. So a really easy way for us to then start sharing more things with our team. However, there's likely going to be times when it's not going to be the most helpful to have give everyone edit access, right? You might have illustrations in there where you only want to make sure that like that copy doesn't get unintentionally edited. So if we go back to our project progress meeting here, let's say that we have our 3 illustrations, but I want to make sure that this components of our HPLC protocol stays as is. So what we can do is actually selectively change the permissions for each of these files so that we give the right permissions to the right people. And so again, we can click on those 3 dots, we can go to share, and then now you see right now everyone has edit access. And so some people have view. So I'm just going to turn all of these people into view access only. And now this way, you'll still have access to this master copy. You can make the changes if needed, and then your colleagues can then make copies of those of that illustration to be able to use in their own work while leaving the original unaffected. And from the other end, if someone shares things with you, they appear down below in the shared with me section. So up top, these are all the things that you've created, and down below, these are things that have been shared with you. And so you can see a folder has been shared with me, and I'm just going to show you what that looks like from the other side. So here we have 2 files, one where I have edit access, where I can click in, I'm able to make changes as necessary. And this is a great way, and this permission setting is great if you all need to collaborate on that same figure. Let's say that you're co-authors on a paper, everyone needs to make edits to this illustration. Having full edit access is more advantageous there. However, if you want to, if there's like, let's say a template that people in the lab should be regularly using, you don't want that template to be accidentally edited, because then that will propagate afterwards. So instead, you can see that this one is view only. And the difference here is that I'm not able to make any edits to it. You can see that my editing panel is gone, and then I'm not even able to click on things within the illustration. But what I can do is go up to File and make a copy for myself. And by making a copy, I can actually create an editable version of it where I can then go in and start changing all of my text. Let's say these are different checkpoints, checkpoint 1, whatever it is. And then we can start adding it into our illustration, but we're able to keep that base as is. So a really great way for us to, share work and share resources with people, and be confident that unnecessary or unintended changes aren't made. Does anyone have any questions about this first tip so far? Any thoughts? We'll take a look into the chat and Q&A here. Looks like we're pretty good so far. But again, if you have any questions that you want that pop into your mind, feel free to ask. Catamayettes are fantastic resources for anything that you have in your mind. So definitely highly encourage those questions. It's also great to see, those gears turning of how we can incorporate these this content or these tips into your own research as well. And one more thing that I want to share with everyone here is that when we create that group right now, you'll see that we had 4 people in our group, and all 4 of those folks actually have access to this folder. Whoops, not this one here, this one. You can see that there's 4 members in our group here. However, there's going to be times when people leave the lab, leave the organization, and you want to make sure that only the right people have access to the folder. So what we can do is actually just go back into the group settings. So back into our profile, and then going into groups. And from here, let's say that Cindy is no longer with the lab. What I can do is just remove them from my group here. And now if I go back out to my gallery, you'll see that if I go back to share, this will automatically update. My lab now only has 3 members in the group. And this allows you to very quickly ensure that only the right people have the correct permissions for your to access that folder. And also anything that anything that the person has put into the folder before they left will actually be in there, it will still stay inside that folder. So you don't have to worry about losing any of that information when the team leaves. A horror story that I've heard before is that someone left the lab, everything was on their laptop, and then they gave the laptop back to IT, and then that laptop was wiped. And then all of the stuff that was on it was just gone. And obviously that's a lot of wasted effort, and it requires whoever's left in the lab to reduplicate a lot of those experiments. So we want to try and help avoid that. So by having these shared folders where everyone can put stuff into, now if people leave, you still have a record of all of those resources, and you don't lose any of that work that was previously created. So highly recommend creating like this shared folder, which acts almost like as a central repository for your illustrations where everyone can contribute into, and would highly recommend setting that up for your own labs. Awesome. And now that, so after that first tip there, the next thing that I want to talk about is how we can create a visual to communicate research quicker and easier. And I'm sure all of us are familiar with this. As a scientist, one of the challenges that we often face is how we can communicate complex ideas efficiently and concisely. And traditionally, the way that we do this is by using text. And so I have this example here with our, something that I think is very familiar to most of us. And this is just a written protocol of how we would conduct a Western blot. And you can see here in this text that we have all of the information that we need. We have the conditions of the experiment, we have the reagents that we use, where we purchased it, any other details that we need to know. All of that is written in text, and it's great for that level of detail. However, for someone that is following this protocol, it can be more difficult. And I use protocols as my example, but it applies to all the ideas and concepts that we share, right? More often than not, we're using text to describe very complex concepts. But if we supplement it with a visual, it actually becomes a lot easier to follow. And a great quote that I think, or a great anecdote from a scientist that we spoke with who runs their own lab, is that a single image can improve your protocol. So for example, if step number 2 is done with a centrifuge, adding an image of a centrifuge into the margin of your protocol can help you, 1, quickly see which piece of equipment is needed for that step, and 2, you can easily see what step you're on. And the third piece is that you know which piece of equipment that you need to use, especially if you have, let's say, 2 centrifuges in the lab. One is meant for this experiment, the other is not. Having that visual cue can remind someone who is just learning the protocol of like which is the correct one to use. And right away here as well, like you can kind of see that in effect, right? You have like this dense wall of text on the left-hand side, and then you have it kind of broken up into more into clear like visual cues on the right hand side. Both I think have like their place in science, but supplementing that text with a quick visual can allow someone to follow this protocol a lot easier. So we are missing a little bit of information. If we wanted to, we can add in all of like the technical details about like the conditions, the reagents into into this white space here. But what I wanted to highlight is that this clearly breaks down the different steps. So as someone that is following a new protocol, this is easier to follow along with. And this is a pre-built template that's available within the library, but likely you'll have a protocol that might be more specific to your lab. And in those cases, we can still use BioRender to quickly create a figure that shows the research, whether it's a protocol or if there's an idea that you want to show, we can use BioRender to build it. And I'm sure a lot of you are familiar with that process already. And for those of you who are a little bit newer, there's some things that I just wanted to review with you first. When you are going to create something from a blank illustration, what I'd recommend is following this one of these simple compositions. And by simple composition, what I mean is like how the information flows in your illustration. So you want it to be as simple as possible, whether it's a left to right in a horizontal line, a top to bottom format in a vertical line. If you need to split it up across two rows, something like a Z shape where you go left to right first and then top to bottom and then left to right again. This is the easiest to follow. And the reason for this is that if you imagine reading a book, particularly in the English language, you always start from that top left corner, and then you work your way from left to right along a line, and then from top to bottom along your page. And having your illustrations follow that pattern is just a more natural way of picking it up. And so would definitely recommend following one of these because this kind of mimics the way that we would typically read and absorb information. And what I wanted to show here, we're going to create a protocol and we're going to put some of those into motion as well. And so let's say for our example today that we wanted to show how we do limited proteolysis coupled with mass spec in our lab. So what we can do is actually just start building this from our blank canvas. So to start, let's say the samples that I work with are these retinal epithelial cells. These are typically hexagonal in shape. So I'm going to look for hexagonal cells to work with. So this one looks pretty good. I'm just going to bring it out into onto my page here. And this is just one example. If you have different cell types or whatever that you're working on in the lab, check out the icon library. And so the icon library is super powerful because it represents over 50,000 different scientific entities. And we do split them up into different cell types, macromolecules, human anatomy, and objects in the lab. And for me, actually, my personal favorite is lab and objects, especially the machinery and tech section. And the reason for that is inside here, there's a lot of icons that represent real equipment that is used in the lab. So the first time I saw this was like the coolest thing. We're able to see like, I could recognize like, hey, like this is the balance that I use in the lab. And it's great for when you're building out these visual protocols or even like showing your experimental apparatus, being able to accurately reflect that in your illustration is super cool. And going back to, that quote that we were talking about, having a visual cue is very powerful for when someone is trying to follow to replicate your experiment. So definitely recommend just reviewing, going through all of the different sections here. And I know folks today, there's a lot of different research areas that are being covered. And so if we go into cell types, for example, we do break that down a little bit further, such as the immune and blood cells for those of us who are in immunology. We can also look for cancer cells, for example. So the library is very broad. Definitely would recommend just kind of going through this to make sure and see what is available for everyone. And going back to our protocol here, let's say we want to continue to build this out a little bit. So I want to indicate that this is my first step. So I'm going to start adding some labels and text here. We're going to call this retinal epithelial cells. And something that you might have noticed recently is that we've incorporated a spell check into BioRender now as well. I'm just going to center this so I can make this box nice in the middle. but you'll see that this word has now been underlined. And what we recently did was ingest both standard English dictionaries as well as scientific dictionaries so that we're able to help flag any typos that occur. And so you can think of this almost as like that final check before you go to present an illustration to make sure that all of those typos are cleaned up. And when the word is flagged, what we can do is right click on that word. And here you'll see suggestions of what we think the word could be. So in this case, it is retinal. However, let's say that I know in science, there's a lot of acronyms that we use. There's a lot of very specialized language, and it's very likely that the dictionaries that we've incorporated don't have that word included in it. So in those cases, if you're certain that this is the word that we're using, this is the correct spelling of it, add it to your personal dictionary. This way, if you need to use that word again, it's no longer going to be flagged as a typo, and you can build out your personal dictionary to reflect the nomenclature that's used in your field of research. But for me, this is definitely a typo. I meant to spell retinal, so I'm going to click that, and so I can easily get that fixed. And what I'm going to do as well, just group this together so I can make duplicates of this after, and then we can start to arrange our elements on our page. And so let's say after we've grown our cells, we actually have this will split off into two paths. One is going to be a treated sample and one's going to be a non-treated sample. And following our simple compositions earlier, we can create this fork path in this horizontal shape. And so we'll make that a branched arrow. And so personally, it took me a little bit of time to create a branched arrow before, and I don't want to have to repeat it again. So what I did was actually just add it to my favorites. And now I can actually reuse the work that I've created before in my current illustration. And to do that, all we need to do is just select the icon or whichever element that we want to save, and then just click on the star on the top right. And now when we go over to our favorites tab on the left-hand side, you can see that my hexagonal cells are there as well. And so this way, if you have complex things that you've created that you don't want to have to make every single time, make it once, add it to your favorites, and when you need it again, just bring it out onto your canvas like so. So it really helps to accelerate, that process of making figures because you're able to reuse a lot of the work that you've done already. And then we can just focus on just constructing the rest of our illustration instead. So let's say that I have my Eppendorf tube here. I'm just going to change the color to make it match. And then we're going to create a duplicate down below. And what we can continue to do as well is, let's say we want to indicate that this sample is going to be our treated sample. So I'm going to grab an arrow like so, and then pull that out here. And one thing that I want to show to everyone as well, like particularly with our arrows, is that when you have arrows that do the same thing, like in this case, showing progression along a protocol, we want to make sure that they look and appear identically, or look and have like the same style. So in this case, you can see that my branched arrow is 2.51 in terms of width. So I want to match that with the arrow that I've just drawn as well. So I'm going to go 2.51 here. And the reason for that is it makes your illustration a lot easier to interpret, and it really helps to make things more efficient because then there's less questions and confusion about what different symbols and elements on your illustration actually mean. By having all of them in the same path, we intuitively know that these are all doing the same thing. And an example that I want to share here as well is with like our text. And so what I wanted to show with the text is that we want to create what we call a text hierarchy. And text hierarchies are how we show importance or order in our illustration. And in this case, the order that I want to show, whoops, I'm going to make this left aligned too, so that it says the same thing. And so let's say that there is text or details that we want to include in this part of our illustration. So what we do is, typically we'll add in our text box. We're going to call this treated, and then we're going to create a duplicate of this. We're going to call this non-treated down below. However, you'll see that one thing that makes it a little bit confusing is that both of these font sizes match the title, the size of the font that we use for each step. However, this is not an overall slide marker. but this is actually like a detail within, and this would be lower on that hierarchy compared to our major umbrella step up above, or that major umbrella label. So what we can do instead is make it a little bit smaller. So this is now 11. And by making this a little bit smaller, what we do is effectively create some separation. So when someone goes to look at our illustration, what we see is that, hey, these are our titles, these are our main headings, and then down below, these are things that fall underneath each of those headings. So it again makes it easier to facilitates that communication and helps to make your illustration more accessible for your colleagues. And then lastly, let's say that after we treat our samples, we're going to create like just a cluster of we have some proteins that we get out of it. And then so I'm just going to create a group here. And then same thing for the bottom, because we did treat this, the only difference between the two is the treatments. We're going to show that we've collected proteins here, and I'm just going to change the color so that we are able to set those apart a little bit. Awesome. And then after this step, if we reconverge into one path, what we can do is then just reuse this arrow again. What I find is actually really handy is that you're able to reuse like a lot of different elements. So again, it just makes it easy to like rebuild things. And you can see that even as like we're talking and putting this together, like how quickly this entire protocol is starting to come to form. And I think that's like the power of being able to use the platform. Like what it allows you to do is create visuals where you might not have been able to. Whoops, just gonna just make it so there's only one arrowhead. But yeah, like being able to like create visual like protocols like really helps like facilitate like those discussions that you would have in the lab. And because a lot of times, like, stories that I've heard from like scientists is that you go in with text, and then partway through the meeting, you're grabbing a white erase marker, and then you're writing on the whiteboard anyways, and you're creating an illustration there. And so instead, you can actually go prepared for these presentations and these discussions by having the visual created beforehand, and then now everyone can work off of that illustration, and it clearly lays out like the ideas that are being communicated. So really powerful here. And, even from like the short 5-10 minutes that we've been working on this illustration, you can already see like it start to come to form. And I'm not going to spend the entire time here making the figure. I just really wanted to highlight like what you can do. And before I forget, one thing that I did want to share with everyone as well, how we talked about favorites earlier, where you're able to favorite things that you've created. A really cool thing that you can do is share icons and whatever things that you've made for yourself with your lab as well. And so what we can do is just hover over the thumbnail, click on the three dots and go to share. And now I can actually share this icon with everyone that's in my lab. And essentially what we're doing here is creating this shared visual language. And again, as you communicate things that are more complex, having that shared visual language can really help to align everyone on align everyone on how you want to represent things. And again, when you go into those discussions, it helps to facilitate them because when you have that conversation, everyone has that same visual in their mind. So I would highly recommend sharing, entities that you've created with your lab, especially if it's some things that are regularly communicated by the group. And like going back to this figure here, I'm just going to wave a magic wand and then transform it into, the final version. And you can see that we were even like throughout this time, we got a third of the way there. So if we were to finish this off, it probably would have just taken us into the 5 to 10 minutes. So really quick for us to create an entire protocol here in the platform. And something that I do want to note as well, right now, this is just dummy data that we've included. However, if you have real control data that you want to include in your figure, just real data in general, you can upload it into your BioRender library. So on the left-hand side under the Uploads tab, you can actually import any sort of image, any supported image type. So PNGs, JPEGs, SVGs, all totally fine. You also have a max of 25 megabytes, so you don't lose any resolution for the figures that you bring in. But what we can do is then upload that data or whichever else, or those images into our BioRender libraries, and then we can just grab it out of our uploads folder, and then just pop it onto our illustration. And so this is a really cool way to pair together the data that we've collected in the lab alongside figures that we're creating in BioRender. And especially with the control data, I think what's really cool is that this allows us to give our the people that are just new to the lab sort of an idea of what to expect in terms of like control values, whether to give them some indication that they have, run the experiment properly, or if they need to go back and like troubleshoot and see what might have been went wrong. So it helps to like add that level of confidence there. Awesome. And I think Kat and Maya have been doing a fantastic job of answering the questions that have been coming in, because I don't see anything that's outstanding right now. But again, if there are any thoughts or questions that you have, feel free to throw the question into the chat there. All right, and moving on to our third tip, which is about using collaboration tools to accelerate feedback and research. So I think a point of friction for us is that we spend a lot of time sending, files back and forth to each other, right? A lot of different versions, and it takes a lot of time. And we create a lot of versions as well. Like oftentimes when I would have a folder that would just say figures, and then underneath would be figure v1.1a, figure v1.1b, and the list goes on. And it was just like a bit of a mess. And then someone had to then consolidate all of those changes together. The comments that were left were either done via email, or it was like verbally told to us by the PI. And it was just a bit of a mess. But instead, what we can do is actually leverage the real-time collaboration feature to be able to edit this figure together in real time. I'm going to show you something that's really cool. So I'm going to actually share this with my colleagues. And so I'm just going to send an invite again. I think Cindy already has access, but I'm going to send her an invite. And then I'm going to send in, oh, perfect, it's been flagged already. And then I'm going to send it to my colleague Eunice as well. So I'm going to send them both an invite. And now what they can do is actually edit this illustration directly. You can kind of think of this as like the Google Docs of figure making. We can all actually get in here and then work on that same illustration together, which really helps to facilitate, that reiteration process when you're creating a figure. And what's also really cool is that I can leave comments for them as well. So let's say that I want to, add numbered steps. I can add that as a comment. And when I add comments as well, we will actually, there's a notification email that's being sent, that's sent out. So you won't miss any comments that are added. And so what I can do here, again, color contrast and call out circles, you can leave the feedback on like areas that you want to have fixed. And when you're all finished with it as well, you can see that, those numbered steps are coming in. I can actually just resolve that comment here and we can just have that check mark. So we know that that's already been addressed. So a really easy way for us, again, to collaborate with our team. And what's really cool, you can actually see that my colleagues are here, like making edits directly to this illustration. They are able to like move things around. And again, we're all working on this figure together at the same time. So really handy way for us to collaborate. And I think that it really helps to facilitate like that process as well, because we're all looking at the same thing. We're all looking at the most update version of the figure. And again, it just helps to make that feedback and reiteration process more streamlined and more efficient. Awesome. And as they finish up here, again, if we wanted to hide any of the comments, we just need to click on this drop-down menu. We can either toggle those email notifications on or off, depending on your preference. Or if we wanted to, if there's like a lot of comments here and it's getting a little distracting, we can also hide all of the comment bubbles and you can see those disappear, but it's still visible to the people that I've left it to, where they can then start to transform this, continue to transform this illustration while I work on it as well. And thanks, Eunice and Cindy, for helping make those changes. You can see like with those little changes and modifications that we're making to the figure, it really transformed it from where it was before to like this final form where it's like nice and clear and things are, all the little details have been addressed too. Awesome. And I know we're coming up on time here. So there's a few things that I wanted to do as we wrap up. So first, just wanted to summarize what we covered, right? We looked at team organization and file management, how we can improve that. We looked at ways that we can create figures to express complex ideas. And then finally, we also looked at how we can collaborate with our team and provide feedback for each other as well. And speaking of feedback, one thing I did want to ask the audience today, if if there is content that you want to see, like after browsing through like the libraries and then seeing like the content and the icons and the templates, if there are areas that you want to see better covered in the library, please let us know. You can either throw it into the chat here. Feel free to list like 1 to 3 topics, or if you think of things afterwards as well, feel free to reach out to our support team. You can click on this big help bubble here, and then go to messages, and then you can start a message and just share some of that feedback with us as well, because we'd love to see where we can continue to add content and further provide value for scientists like yourselves who use the platform. Awesome. And to give you some ideas on where you can use BioRender as well, this is just like common ways that we see BioRender being used, but essentially you can use it across your entire scientific journey. Anywhere that you need to create an illustration, you can use BioRender to create that visual for you, anywhere from like the ideation and brainstorming phases all the way up to your final project deliverables. You can create all of those illustrations within the platform, and our goal is to give you something where really it's only your imagination that limits what you can create. And as we wrap up here, a few sort of PSAs. First, we are offering a free trial that includes all of our premium features. This will get you access to the slides, unlimited figure storage, and access to our full icon library. And for everyone that attended today and everyone who registered, there will be a coupon code that's going to be sent out for you for a free 30-day trial, as opposed to our standard 14-day trial. So it's going to be a unique code. But if you already have a premium account, don't worry. You can actually share the code with your friend or a colleague if they're interested in trying BioRender as well, so that they can get that extended trial. And just as a heads up, the code does expire in 30 days, so remember to use it before then. And as we start to wrap up here, I just wanted to just launch one last poll, just asking about your feedback here. Oops, let me just end this one and then launch this second one here. Just asking how you found this content. Like, we're always looking to improve and provide content that is most valuable for scientists. So if you have any thoughts that you want to share, just let us know, like how helpful this was, whether you'd recommend it to a colleague. So on your way out, feel free to just drop an answer there. I just launched the poll, so you should see it soon. If you have any questions, I'm happy to stick around for a couple minutes if anyone has any last thoughts on their mind. If not, again, if you have questions that pop up afterwards, feel free to reach out to us via the help chat. And for anyone that joined late, we will be sending a recording out to everyone, so keep an eye out for that there. And again, wanted to thank everyone for taking the time to join me. I know there was a lot of activity in the chat and Q&A, so I want to thank everyone for their thoughts, their questions. Really, again, it is great to see those wheels turning in everyone's head about how they can incorporate the platform into their work. Again, thank you for your time today, and happy illustrating, everyone. Awesome. Thanks so much for the kind words, everyone. All right, if there are no last questions here, I'm going to go ahead and close down the webinar. Thanks again, everyone, and hope you all have a wonderful rest of your day wherever you are in the world. Take care, everyone.